Today I am going to talk about the reabsorption at Loop of Henle. So this is the sixth lecture about the urinary system. Loop of Henle has three functional segments. That is the thin descending part of the Loop of Henle, thin ascending part of the Loop of Henle, and thick ascending limb of Loop of Henle. So these three regions, especially the thin limb as well as thick limb, they have functional differentiation. This thin loop of Henle has thin epithelial membrane. The membrane of this part of the nephron is made up of epithelial cells which are very thin. These thin epithelial cells has got limited number or less number of mitochondria, and also their metabolic rate is quite low. Because this is related to their function, the reabsorption happening in this area is mainly by passive transport, where there is no need of any energy. Whereas in the case of thick ascending limb, there is a lot of active transport mechanisms happening. As a result, the metabolic rates of the epithelial cells here is quite high. So for this high metabolic rate, there are a lot of mitochondria which produce a lot of ATP. As this active transport need enormous amount of energy which is supplied by the ATP. So the functional and structural differences are there in these two areas. So loop of Henle, as I said before, the different part of the loop of Henle has differential functions, and the descending loop of Henle, the thin loop of Henle, the descending part has high permeability to water. Their tight junctions are highly permeable to water, and also there are special mechanism for reabsorption of water. As a result, 20% of the water reabsorption takes place here. So recent research suggests that there are certain proteins which acts as channels in this descending loop of Henle, thin segment of the loop of Henle. Those proteins have sulfidyl groups, and these sulfidyl groups helps in the opening or closing of these channels, through which water can move into the cell and from there into the interstitial space. And also the presence of aquaporin is a water channel. This acts as a marker for this descending thin loop of Henle, because these aquaporins are not present in the short loop of Henle. That is, the nephrons which has a short loop of Henle is not having this aquaporin. Whereas in the case of nephrons with the long loop of Henle, there are aquaporins. So these aquaporins are supposed to play an important role in the water transport, and also there is a little bit of secretion of urea into this part of the tubule, which help in the urea excretion. So that was a thin descending loop of thin limb of loop of Henle. In the case of thick ascending loop of Henle, the reabsorption is comparatively nil. Its tight junction, the tight junction of the cells in the thin ascending loop of Henle is very tight and impermeable to water. There is no water reabsorption or any solute reabsorption in normal case. But in the case of thick ascending loop of Henle, the thick part of the loop of Henle, which is ascending part, the reabsorption is quite high. So it plays a major role in the kidney function. This is an anatomical and functional position, central position in the kidney renal function. Its critical roles include maintaining extracellular fluid volume by absorbing many of the solutes. It controls the extracellular fluid volume, and it also helps in the urine concentration mechanism, and calcium and magnesium homeostasis. A lot of calcium and magnesium is absorbed at this thick ascending part of the loop of Henle, and it also plays a role in the acid-base balance by absorbing bicarbonate or excreting or secreting or removing ammonia, and also it controls the urinary protein excretion. So thick ascending limb of loop of Henle, as I said before, it plays a major part in the reabsorption of sodium, chloride and potassium ions. A quarter of these ions are absorbed here, and also it is a major location of magnesium and calcium reabsorption, and we are going to see the mechanism in the later slides. So thick ascending loop of Henle, there is a large reabsorption of solutes. There is no reabsorption of water. As I said before, the tight junctions are impermeable to water. As a result, no water reabsorption at this part, but a lot of solutes are reabsorbed. This causes a dilution of the fluid in the lumen. So this part is, or because of that, this part is called diluting segment of the nephron. So solute reabsorption, there is no water reabsorption. As a result, water content increases. That is why it is called diluting segment. Because when it enters, the fluid enters the proximal convoluted tubule, the osmolarity is 300 milliosmol. As it moves down into the loop of Henle, the osmolarity increases at the, while it is passing through the descending loop of Henle, it is 300 to 400, 600, 900 and 1200. Later what happens, there is very little reabsorption here, thin ascending loop of Henle, but in this thick ascending loop of Henle, there is a lot of reabsorption of solute. As a result, the osmolarity decreases to 200 or up to 100. That is a lot of water there. The solute concentration is quite low. So that is why it is called diluting segment. And as in the case of proximal convoluted tubule, here also the reabsorption, the active reabsorption of most of the molecules like chloride or potassium depends upon the reabsorption of sodium. It is powered by the basolateral sodium pump, sodium-potassium pump. So the reabsorption of chloride and potassium is molecularly related to reabsorption of sodium, as in the case of proximal convoluted tubule. And also, this apical membrane, the membrane facing the lumen of the tubule, is permeable to, it has an inherent permeability, some slight permeability for potassium ions. As a result, potassium ions move from the cell into the lumen of the tubule. So this increases the potential, electric potential of the luminal fluid slightly to a positive side. So the positive potential is increased. And this increase in the positive potential causes the movement of positive ions like magnesium, calcium, sodium, potassium through the paracellular route into the interstitial space. From the luminal fluid to the interstitial space through paracellular route. This is happening because of this slight positive potential. That positive potential is generated by the potassium leakage from the epithelial cells, because the epithelial cells have an inherent slight permeability for potassium ions. And also there are the sodium Na plus H plus transporter, NHE3, its name is NHE3, and that plays a major role in the acid-base balance, because hydrogen ions are excreted where sodium is reabsorbed. All the reabsorbed sodium, here also the sodium is reabsorbed, is taken or pumped out of the cell by the sodium potassium pump on the basal side, basolateral side of the epithelial cells. And this sodium reabsorption powers the reabsorption of the chloride as well as potassium ions. So sodium is pumped out here by this sodium potassium ATPase sodium pump. As a result, the cytoplasmic sodium level decreases. This generates an electrochemical equivalent, because low sodium here, high sodium here. As a result, sodium moves into the cell because of this electrochemical gradient. And this gradient is generated by this active transport, which is primary active transport. And this primary active transport actuates this sodium reabsorption. So this is a secondary active transport where there is no use of energy, direct use of energy. But the energy used here potentiate or power the reabsorption of sodium. When the sodium is moving in, it causes the movement of chloride ions as well as potassium ions. So this chloride and potassium ion reabsorption is molecularly related to the sodium reabsorption. And also the potassium secretion due to the permeability of this membrane increases the positive potential here, which push the other positive ions from this luminal fluid into the interstitial fluid through the paracellular route. And as I said before, the bicarbonate absorption the thick ascending loop of Henle plays a major role in bicarbonate reabsorption. Around 15% of bicarbonate is reabsorbed at the thick ascending loop of Henle, and this is facilitated by the Na plus H plus exchanger NHE3. So this bicarbonate absorption is made possible by the presence of this exchanger, which causes the secretion of H plus into the luminal fluid. And when these H plus ions are exiting out of the cell and going into the or secreted into the luminal fluid, when one H plus ion is secreted into the luminal fluid, one HCO3 minus ion is pushed or moved into the interstitial fluid. So 1-to-1 relation, one H plus ion secretion, one HCO3 minus ion reabsorption. So this is what happens in the case of bicarbonate reabsorption. So H plus ion is secreted into the luminal fluid by the Na plus H plus exchanger, NHE3 exchanger. One sodium ion is going in, one H plus ion is going out. So it is exchanging, so it is electroneutral. One going in, one going out, both positive ions. And this H plus ion combines with SCO3 minus ions. This SCO3 minus ion plus H plus ion forms carbonic acid, H2CO3. This carbonic acid, as in the case of proximal convoluted tubule, cannot move through the plasma membrane. As a result, and in the presence of carbonic anhydrase enzyme, this carbonic acid split into carbon dioxide and water, and carbon dioxide being lipid soluble can easily pass through the cell membrane, and the carbon dioxide reaches the cytoplasm. There it combines with water, forming again carbonic acid in presence of the carbonic anhydrase enzyme. This carbonic acid, which is highly unstable, splits into two, not as carbon dioxide or water, but here it is split into H plus ions and HCO3 minus ions. So this H plus ion is secreted into the lumen, and which is used for the reabsorption of further molecules of HCO3 minus. And the HCO3 minus ions produced here it is moving into the or absorbed into the interstitial space by different mechanisms. One mechanism is this K plus Na plus exchanger or K plus HCO3 co-transporter. This is co-transporter, not exchanger. Co-transport. Potassium ions are moving out, HCO3 minus ions are also moving out. So this co-transporter plays a role. Then this HCO3 Cl minus exchanger. When Cl minus is moving in, chloride ions are moving in, and bicarbonate ions are moving out. So it's bicarbonate chloride ion exchanger. Then this potassium channel helps in the movement of chloride ions to this outside the cell, and this Na plus HCO3 minus transporter is also present on the basal border, and this is highly expressed in the cells of the thick ascending loop of Henle. But it is not helping for the exit of bicarbonate from the cell, but it causes entry of bicarbonate into the cell. So its function is believed to be entry of bicarbonate ions into the cell as well as entry of sodium ions, and they are highly expressed in this basal border of the epithelial cells in the thick ascending loop of Henle, and their name is NBC-N1 transporter. And this Na plus H plus transporter that has also got an important role in bicarbonate reabsorption, because H plus ions are needed for the bicarbonate reabsorption from the luminal fluid. Here H plus is going in and Na plus is going out. That is an exchanger. Na plus H plus exchanger. That is also help in the movement of H plus ions into the cell, which will later move into the luminal fluid and cause the reabsorption of bicarbonate ions. And this bicarbonate absorption is upregulated in acidosis and downregulated in alkalosis. And most of this bicarbonate reabsorption happens through these transporters, and especially these transporters, this Na plus H plus or Na plus K plus SO3 minus transporter is upregulated during this process, and it increases the bicarbonate reabsorption. And ammonium transport. Ammonium ions are produced or synthesized or created in the proximal convoluted tubule in response to acidosis. When acidosis, body acidosis is there, ammonia ions are produced from glutamate in the proximal convoluted tubule. That ammonia ions are secreted into the epithelial cells of the proximal convoluted tubules into the tubular lumen in the proximal convoluted tubule, but there is no reabsorption of ammonia ions in the proximal convoluted tubule. So that ammonium which was produced at the proximal convoluted tubule moved till it reaches the thick part of the ascending loop of Henle. So where it is reabsorbed. So how is it reabsorbed? Here also this Na plus ammonium bound exchanger or Na plus K plus exchanger plays a major role. Here from this cellular lumen, ammonia enters into the cell through the Na plus K plus exchanger or Na plus K plus NH4 plus exchanger. That is, K plus is going out, ammonium ions are getting in. And also this symporter, which is a Na plus K plus 12 minus symporter. So it helps in the movement of these ions into the cell in the same direction. That is why it is called symporter, and it helps in the absorption of ammonium ions, because the ionic radius of potassium ion and ammonium ions are same. As a result, ammonia ions can move through this transporter, this symporter. So that plays a role. A major role in the reabsorption of ammonium from the tubular lumen into the cell. And also this potassium channel. Here ammonium is reabsorbed. But in normal case, the role of these potassium channels as well as this extracellular transport, paracellular transport, ammonia is also taken into the peritubular space or interstitial space by paracellular transport. But in the normal case, these two, this paracellular transport and ammonia, this potassium exchange, potassium channel, the ammonia absorption through the potassium channel is not happening in normal physiological conditions. But when there is high acidosis, this can come into play. So once the ammonia ions enter into the cell, it can either go to the peritubular or interstitial space through this Na plus K plus exchanger, or it can be split into ammonia and H plus ions. This ammonia is moving into the peritubular space. And also there is one more co-transport mechanism, ammonium chloride ion co-transporter. So here ammonia ions as well as chloride ions are transported into the peritubular space. So these are the transporters and exchanges taking part in the removal or reabsorption of ammonium in the epithelial cells of ascending loop of Henle. So this picture shows a summary of what is happening in the loop of Henle and how it is affecting the osmolarity. So when the fluid is moving through the descending thin loop of Henle, lot of water is reabsorbed. As the solute reabsorption is very minimum, almost nil. As a result, the concentration of the solution increases, the osmolarity increases from 400 milliosmol to 1200 milliosmol. And it goes up, there is not much absorption, reabsorption at the ascending part of the thin loop of Henle, thin ascending loop of Henle, but when it reaches the thick part of the loop of Henle, the ascending loop of Henle, a lot of sodium chloride is reabsorbed, calcium is reabsorbed, magnesium is reabsorbed, bicarbonate ions are also reabsorbed. This calcium and magnesium reabsorption happens in the thick epithelial cells of the ascending loop of Henle through a paracellular route. That is, lot of potassium ions are secreted, which causes the movement of positive ions. And around 60% of magnesium reabsorption happens here. You may remember at the proximal convoluted tubule, where the magnesium was reabsorbed before, the reabsorption was only 10 to 30%. So a lot of magnesium remains in the fluid, and out of that, a majority of magnesium is reabsorbed. 60% is reabsorbed and 30% at the proximal convoluted tubule. So 90% or more of total magnesium is reabsorbed. So because of this reabsorption of the solutes, chloride ions are also reabsorbed. The fluid becomes very dilute. There is no reabsorption of water. So as it reaches towards the end of the thick ascending loop of Henle, the osmolarity decreases to less than 200. Maybe it can reach up to 200 milliosmoles. So that is why it is called the diluting segment. So in this way, the thick part of the ascending loop of Henle plays a major role. There is a central role in the renal physiology. Thank you.